Yeah,
I think recording has been started.
okay, good, good. Let's see where we get. I'm going to go back and start where we just give you a quick recap. We were looking at virus classification and how viruses are named, and I just gave you some examples that they could be named based on the disease, the scientists, the geographic locations, or Latin or Greek names. Subsequently, we scientists try to classify it based on the host range, the organs, the like if liver is affected, then it'll be called hepatitis viruses. Then blood-borne viruses, that is the route of transmission, all these things. And then I explained to you what are the differences in the classification of viruses. There is nothing about the family level, which is very unusual. And we still have not yet adopted binomial nomenclature for viruses. I gave you some other examples. Then we looked at Baltimore classification, where virus mRNA synthesis is the central schema for classification. And I went through the slide and I just gave you some examples. These are the different hosts. And then we'll try to take next 20-30 minutes and finish this class. And what I'm going to try and do is to try and tell you how within a virus there is more further classification. Okay, that's what I'm going to try and tell you in today's class. So anybody wants to guess what are the classifications within a virus based on whatever we've done so far, or you've heard about, or whatever you know from, let's say, news, reading newspapers, how a given virus can be classified further?
So like maybe influenza virus has been classified into H1N1 and H3N2 likewise based on
Okay,
the
that's
genome.
correct. They're called types of influenza virus. That's correct. They are types of influenza virus. That's correct. Maybe we can also discuss that. I have those lectures separately for influenza virus, but perhaps we can try and discuss that in today's class because you brought it up. I'm just going to write it down here. Influenza virus. We'll try to discuss that. Okay, what else do you know about classification of a given virus apart from influenza virus types?
So, on the basis of the mutations that take place, like in the case of COVID, we have different mutations, and that is how we've classified them.
And what do we call them?
So strains.
No. For COVID, what do we call them? What are these?
Variants. Variants.
Yeah, they're called variants of interest or variants of concern, right? That's what they're called for COVID or SARS-CoV-2. That's what they're called. Anything else that you know of? No? Okay, so we'll try to look at it in today's class. So mostly, many viruses are classified into genotypes and subtypes. I'll tell you what genotypes and subtypes are. Name suggests everything to do with this comes from the genome, and that's why it's called genotype, which means what is different between these genotypes and subtypes? Guess what is different? What makes a genotype a genotype? Let's say we have genotypes 1, 2, 3, 4, 5, 6. What makes genotype 1 1 and genotype 6 is called 6? What is the underlying reason?
Difference
Changing
between
their
genetic
genome.
sequences.
That's correct. Everything has to do with differences in the genomic sequences, and that's what I have here. I'm going to try and explain this to you a little bit more. So if you start reading about viruses, or if you work on viruses, what you'll see is you'll often come across phylogenetic trees. And this is one type of phylogenetic tree. And what you can see is like, if you look at it, this is what it looks like. But you can see wherever I've indicated the circles, they form a group of sequences. And if you look at this, everything within a circle is generally, let's say this is genotype 1. This will be genotype 3. This will be genotype 2. This will be genotype 6, and so on. Right? So why are they called? Why are all these sequences called genotype 6? Because they are quite different and they branch off here from the other closest genotypes, which are genotype 4 that goes here, or this branch here, genotype 3, they're different from everything. They are a completely different bunch of sequences from this group, this group, this group, and this group. So that's why they branch off from the tree. And subsequently, if you see, there are further smaller branches. Now, these sequences, each line is going to be one strain or one sample. If you see, maybe there are like 30, 40 lines in this, and they all bunch up together. They have similarity and differences. They are similar enough to bunch together, but they are different enough, for example, from this bunch here, 6E, they're different enough from this bunch. They're different from this one. They're different from this one, that they bunch up together, but they're different enough from others. Okay, so now that's what makes genotypes. And 6A, 6U, 6E, 6C, for example, all these are called subtypes. 6 is the genotype, and the A, 6B, 6C, these are the subtypes. Now, how are they different? Okay, just going to erase off all of these and try and tell you a little bit about how they could be different. Now, these are usually arbitrarily classified. What do I mean by arbitrary classification? For example, when we, for example, in hepatitis C virus, in which I think this might be HEV. I think this is HCV, I think, if I'm right. But doesn't matter what this tree is from. I'll give you an example. For hepatitis C virus, if you were to classify, for example, let's say there is 30% difference in the sequence between two sequences, any two sequences you analyze, they will form usually different genotypes. Now you take 5 sequences from a genotype, and then you look at them. Let's say there are at least 12% difference between, just an example, something like this is used. These numbers are not important. If the sequence of this strain or this sample is different from this sample by 12%, then they will form subtypes, which will be 6A, 6B, 6C, 6D, etc. But like 4 and 6 genotypes, they would be different by at least 30%. But within a genotype, the subtypes may be different by 12%. Again, these numbers don't mean anything. These numbers are just an example. But something like this is used for HCV. For another virus, it could be 15% and 6% for genotypes and subtypes respectively. But so it could be very, very different for each virus. So you don't have to, the number is not important. The concept is important. The concept is that like different genotypes are more divergent than different subtypes, because within a genotype you have subtypes. Anybody has any questions about or did not understand this well?
Yes, sir. I have a question.
Yeah, sure, go ahead.
Sir, the thing is that I know about like this thing, this type of classification about a species of bacterial species also happens. Like we consider that if 70% of the genome is same, then the bacteria is of same strain. But like I was
that is not true. Bacteria, same bacteria don't have 70% similarity. They have much higher similarity. And see, bacteria has genus and species. And usually bacteria, in general, bacteria don't have genotypes or subtypes. There could be few bacteria which have, mostly they don't have. And 70% is not the cutoff for species
I guess
identification or
70% or
strains.
species or 80%, but not more than that, sir. I have read in book actually.
So what
Yeah,
I would
like
recommend.
bacterial species are not defined yet, something like that.
Say that again. Say that again.
Like a species of bacteria are not exactly defined in that way. Like it is defined for mammalians and etc.
Mammalians. What is
Like species in mammalians and other animals are like if they can reproduce with by mating with each other, then we consider that they are the same species. If they cannot reproduce, we consider that they are not of same species. But that doesn't perhaps that kind of definition doesn't apply for bacteria. So we have the genetic similarity kind of definition for bacteria, defining bacterial species.
I think Ratnesh, you're getting into. See, I'll give you an example for defining species among eukaryotes, for example. I'm sure many of you are already aware that, for example, humans and chimpanzees, our closest ancestors, are different only by 4%. Okay, this is one thing. It's very different. You can't compare humans or bacteria with viruses. The reason being viruses evolve much faster. Why do viruses evolve much faster? For most organisms in the world, the rate at which they evolve depends on one particular thing. Anybody wants to make a guess?
The rate of mutation.
Rate of mutation is one thing, but rate of mutation, it becomes dependent on something else. What is that? What is the one thing that will determine? Huh?
The
So viruses
complexity.
like replicate at very rapid rate, and also mutations occurs due to lack of proofreading. So
Okay, so very good. Actually, whoever said that the rate of replication may matter. See, see how many times do humans have offsprings in their lifetime? They have one or two offsprings at best these days. So one human being or two human beings together produce one or two offsprings on average. Let's say, just an example. It may not be accurate data, but just an example. But one virus goes in, and in one cycle it can produce like 100,000 viruses. Now those 100,000 viruses can, at least few of them, infect cells successfully, and they will produce 100,000 viruses. So the evolution of the number of times a a virus replicates in a host, in a single host. Let's say somebody who gets infected just for 3 or 5 days, the amount of replication that happens is comparable to millions of years for eukaryotic organisms. Okay, and bacteria double, viruses go exponentially, so they work differently. So again, defining a species at a eukaryotic level is not necessarily by whether they can actually produce offsprings, like you said. That is also not correct. There are animals, actually different animals. A lion and a tiger can have offsprings. Of course, they are different species, and we know that, but they can have offsprings. That is also not how you define a species. But why are we getting into that? We are talking about viruses, right?
But they can, if they can produce offspring, their offspring may be sterile and not able to reproduce. Like moles and are produced by donkeys and horses, something like that.
Yeah, and now science has shown that they can also reproduce. There are some challenges, but they can reproduce. Give me a second. Hello.
Hello. Yeah, good morning, sir.
Good morning.
Sir, I'm calling from Dr. Kalyavani. अभी next turn आपका है, तो आप please be ready. I'll call you again to join the meeting. It will take I think 10 to 20 minutes.
Can you hear me?
Yes.
Yes, sir.
Yes, sir.
Okay, so okay, let's not get into that loop. There is now new literature. Generally, these animals are sterile, but they can also reproduce. But see, we cannot keep on arguing about how is a species defined in eukaryotes. It's simply not defined by those parameters. It's defined by genetic parameters only as of today. Okay, so genetics is everything in terms of defining a species. It's all based on the genome sequence of animals, genome sequence of bacteria, genome sequence of viruses, and that's what we are looking at. So it's not whether they can have an offspring together or not. Okay, so let's leave that aside. Now, I have to join another meeting in 10 minutes. Let me do this in the best way we can. Give me a second, and we'll finish today's lecture as best as we can. Actually, this is spillover from the last class, and I think okay. Thankfully, my computer is moving. Okay, so that is, you have genotypes, which are broader category, and within genotypes you could have subtypes. Okay, so that is the general classification of viruses. Now, within a subtype, you could have something called a quasi-species. Now, what is a quasi-species? Like you can see it from this cartoon also. Maybe this one is a good example. Like this one is a good example. These are good examples. They are the same subtype, but they have differences in sequence. Now, in the same example I gave you, perhaps we can do 30% different will make it genotype. At least 12% will make it subtype. Anything less than 12% different, will make it quasi-species. That's what it's going to make. So that will be, examples will be what I've circled here. These are going to be genotypes. So these numbers, again, don't get fixated on these numbers. They could change from virus to virus. Now, within this, you can see that these are the same subtype, but they don't have identical sequences. They have differences in sequence. So that is called quasi-species, and that's what is represented in this cartoon also. So these are genotypes, and they branch off into subtypes. Within subtypes, you can have quasi-species. You will have this for pretty much most viruses. All viruses have quasi-species. That means if you put one pure form of virus, let's say you take one virus and put it into my body. One virus has only one sequence, and all of us are aware. And if that virus replicates in my body, it will lead to something like this. It will lead to different sequences. All the viruses produced in my body are not going to be the same sequence. They're not going to have the same sequence. They're going to have mutations. Some will have 5 mutations, some will have 10 mutations, some will have 20 mutations, and that contributes to quasi-species, and all viruses have quasi-species. Now, this is just an example. So if you put that one virus into my body, and these are the mutations that occurred when that one virus replicates in my body, you can, if you do bioinformatics analysis, you can combine that these sequences look alike, these sequences look alike, these sequences look alike, and these two look alike, and this one is not a match to anything else. So you can also quantify the different types of quasi-species that are present in a given infected individuals. Or in other words, these quasi-species usually emerge due to errors in replication and the rate at which these viruses replicate. Okay, so that's what this is about. Then there is also one more thing called virus serotypes, a very important concept, a very simple concept, but important to understand. So anything that changes in the genome may affect the virus serotype, may not change the virus serotype. What do I mean by that? So many of us know dengue very well because we live in ITD campus and it's a hotspot for dengue. And dengue viruses come in 4 different formats at least. 1, 2, okay, it goes like this. Okay, this is first one, second one, third one, and fourth one. What is so noticeable about this? The spikes on the surface, they look different, right? That's the difference between the four serotypes. So when a genotype change affects important epitopes on the virus in a manner that the antibody produced, give me a second, the antibody produced against this virus will look like this. The antibody produced against this virus will look like this. The antibody produced against this virus will look like this. And this virus will look like this. Just a crude form of representation. It doesn't actually, all of them look like Y, but they have subtle differences. But you understand that this virus will elicit a different response from this one and this one and this one. So what this means is if you get infected with dengue virus serotype 1, it may not offer protection against the other one or the other one or the other one, because the antigenic epitopes are so different that the antibodies produced will also be different and they may not cross protect. So when you say there are virus serotypes, for example, in dengue serotypes 1, 2, 3, 4, there is difference in the genome that makes one call them different serotypes. They're definitely different genotypes, but they're called different serotypes because they elicit a different serology or they elicit a different antibody response. They're so different in terms of important virus epitopes or virus proteins that when they get into a body, the antibody response and the immune response they elicit is very different. They may not be even cross protection. So that assigns viruses serotype status. Dengue
So I
virus
guess
has
in
go ahead.
So I guess in dengue, it also I'd read about this thing called antibody dependent enhancement
That's
that
correct.
happens in
That's
dengue.
correct. That's very correct. We'll come to it. So there is something called ADE or antibody dependent enhancement in dengue that is related to the differences in serotype, and it happens on reinfection. And I'll talk about it more. It enhances the pathogenicity of the virus if you've already been exposed. Or in other words, if you get dengue for the second time over, it worsens the symptoms. We'll talk about it later. That is true. That is related to the serotypes. Absolutely correct. So I don't know what I have here at the bottom of the slide. I'm just going to my computer. Okay. Okay. So what I have here is it just says that different serotypes are associated with different responses linked to different antigens. That's basically what I said. And this will be the last one. I think I'll close with this. So, for example, one of you said variants of concern or variants of interest for SARS-CoV-2. They're labeled as variants of concern or variants of interest based on their ability to be more transmissible, cause more severe disease. More transmissible, best example is Omicron. This one is perhaps Delta as an example of severe disease. Reduced protection by vaccines, again, Omicron is a good example. Escape diagnosis, Omicron is missed by some PCRs, so Omicron is an example. But I'm not saying others are not. So most of them had at least increased transmissibility, increased severity of disease. Many of them also qualify for this. This one, particularly Omicron, qualifies for this. So they're called variants of concern, which means they have a specific set of mutations, not necessarily one mutation, usually a specific set of mutations that makes them replicate faster or transmit more easily or latch on to the receptor more easily or cause more disease or escape neutralization or become non-detectable by standard tests. So then they're called variants of concern or variants of interest. So I think what the summary of this is that they have a tangibly different phenotype, something that you can recognize as they have a different property. As a bunch of viruses with the mutations, with a group of mutations, they have a different property. Then you call them variants of concern or variants of interest. I think that's all I have. Anybody has any questions? We'll do, because I have been asked to join another meeting in a few minutes. I have to be ready and join the other meeting. So tell me if you guys have any questions. We can do it, and then we close for today.
So,
Yeah, go ahead.
you'd mentioned that there's different thresholds for different viruses when defining genotypes, subtypes, and quasi-species. So
That's
what
correct.
is that threshold based on?
Okay, that threshold is based on actually drawing trees like this. You can see that this is coming off at a different branch, right? So trees can be drawn in different ways. This is called an unrooted tree, and there are also rooted trees. Regardless of whatever you're using, for example, I gave you these examples for HCV. For HBV, different genotypes are only 8% different. Uh, what are they based on? They are based on how these branches occur in the tree when you do a phylogenetic tree, and then you calculate an average percentage, and then you make some rules. And usually for RNA viruses, it's much higher, and for DNA viruses, it's much more tighter because they vary less. That's what it's based on, and it's more or less usually an arbitrary number, but based on the branches like I've shown here.
Got it, sir. Thank you.
Sure. Anybody else has any other questions?
Yes, sir. So will it be like that these genotypes will have different epitopes like serotypes are having, as they are very different in the genome sequence?
Yeah, so a change in the genotype may or may not result in a different serotype, may or may not. But a change in the genome will change the genotype, right? I mean, will affect the genotype. I will not say change the genotype. A change in the genome makes it different genetically, but it may or may not affect the serotype, because to affect the serotype, you may need a large number of changes. Does
Okay.
that make sense?
Yes, sir.
No? Okay. Okay, I think we'll close for today because
Sir,
go ahead.
sir, I have doubts about bacterial species. So, sir, I have questions. Sir, can I email you so you can answer?
Yeah, you can also call me or email me. You have my number.
Okay, sir. Yes, sir. Thank
Okay,
you, sir.
but after 12 this afternoon, we'll do it. Okay.
Okay, sir. Okay.
No problem. Okay, bye. Thanks.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, guys.
Thank you, sir.